Good afternoon, everyone, and thank you for such a warm welcome, Colleen. It is my distinct pleasure to have this, what I hope will be a conversation with you, because as many of you know, the COVID vaccine trials have raised a lot of questions, and we are in the process of finding some of these answers. But to really understand why so many of us had questions, because we didn't really have a lot of questions about many of the other interventions till date, has been the state of affairs, the current state of affairs that we find ourselves in. So I'd like to give a touch briefly on what the NIH vaccine strategy has been, and that includes creation of the Coronavirus Prevention Network that Colleen mentioned, and Operation Warp Speed. And the terminology Operation Warp Speed makes everyone nervous because if people think, rightfully so, that, oh my God, they are just speeding up the processes of vaccine without really pausing to understand the impact and safety. And I hope this talk will help get us to some of the checks and balances we have in place and what the different studies are and the multiple options that you might have. Because ultimately, I will be sharing key data from 4 of the studies that are currently in phase 3 that will likely be going for an emergency use authorization indication to the FDA in the next weeks to months. There we go. So what is the CO-VPN? The Coronavirus Prevention Network, and the website there, if people are interested, is at the bottom of the slide. It's essentially funded by the National Institute of Allergy and Immunology and Infectious Diseases, which is a branch of the NIH. There are currently approximately 200 sites across, globally, and the network has 2 key initiatives. One is looking at effective vaccines that are safe and effective, and looking at the role of monoclonal antibodies to treat and prevent new infections. They were able to rapidly deploy these sites because the NIH leveraged on already funded existing clinical research sites that were doing other infectious diseases research. So essentially, they combined the HIV vaccine trials network, the HIV prevention trials network, the infectious diseases clinical research consortium, and the AIDS clinical trials group. And just so you know, our site has been funded for over 7 to 15 years to do both AIDS clinical trials group work and HPTN work already. Therefore, we were ready to take on these new challenges. So what does Operation Warp Speed refer to? So the ultimate goal is to really help fund a way for us to safely come out of this pandemic. So how do we get there? The overarching goal is to try to see initially, at least to deliver 300 million doses of safe and effective vaccines, hopefully by January 2021. And why are vaccines important? Essentially, when we look at the successful eradication of many viral infections, while antivirals have been part of the solution, vaccine and having a safe and effective vaccine is probably the most key component to help us emerge from that. When we look at the eradication of smallpox or polio, it really has been effective vaccination strategies that were conducted appropriately and safely that helped us overcome those diseases. This was a joint initiative by the Department of Health and Human Services, the Center for Disease Control, the NIH, BARDA, which stands for Biomedical Advanced Research and Development Authority, and the Department of Defense. It also received support from a number of other federal agencies and numerous pharmaceutical companies. So essentially, we've taken the best scientific minds and partnered with industry partners along with implementation scientists to try to help us find numerous ways to help address the pandemic. And what has been the funding timeline? I'm not going to get into, I'm not going to read these slides, but essentially over the past few months, the NIH and Operation Warp Speed has either performed the work themselves or partnered in conjunction with numerous industry partners, ways to help fund multiple different COVID vaccine studies. Once the vaccine candidates appeared to be promising, the government helped support the manufacturing process in a hope to expedite the timeline, with the understanding that none of these vaccines would actually be deployed in the market until they have been shown to be safe and effective. And the funding, the Congress funded a total of $10 billion in March of this year. So that's a lot of money to help support not only clinical research, but also the CARES funding that helped fund a lot of the COVID testing initiatives in this country. In addition to ensure that everyone conducting the studies were doing the studies, in parallel. Traditionally, what has happened with many other clinical research studies is you have a company A studying the impact of one drug, and in isolation. And drug B will be compared by another company with a completely different placebo in such a way that you really couldn't compare the two drugs from two different companies in the same context. So it would make it very difficult to extrapolate the information in a meaningful manner. So what the NIH and the COVID supported infrastructure has done is that it has harmonized the efficacy trials. And what do I mean by that? The clinical trials, the various trial designs, and launching and opening of those studies are done sequentially. So what that means is the multiple different studies of various COVID vaccines, even though they are done by different companies, they have similar clinical trial designs. They have similar patient populations, and they have similar safety tracking. So, and what that helps us do, we hope, is when the results become available, we will be in a somewhat meaningful way compare the results, even though it's not a head-to-head comparison. The clinical trial data will be somewhat comparable. In addition, the endpoints, and what are the endpoints are really the primary efficacy. So when we do a COVID vaccine study, we want to make sure that the cases that are counted similarly across the different vaccine studies. It makes no sense to have one company study the number of symptomatic COVID cases at the endpoint. Another company will get asymptomatic COVID cases at the endpoint, because then you can't really compare the two. By ensuring that the endpoints are the same between the different studies, we are able to compare the data equally. We have also standardized the correlates of protection. So what do I mean by that? In order to prove efficacy, you are looking at antibody titers and the number of cases. Different labs can do differential antibody testing, and so you get different results. And so one's apple, the other's oranges. You can't really compare apples to oranges. By centralizing a lot of these laboratory processing techniques in a centralized lab, you make the data much more standardized. And you have the labs and the sites all collaborating together. And the last, and what I think is the most critical key component, is it's an NIAID managed DSMB. A DSMB stands for Data Safety and Monitoring Board. This is an independent committee that is separate and completely separate from both NIH and separate from pharma. Their sole job is to look at the clinical trial data in an unblinded manner and look to see the efficacy of the vaccine. So they will be looking at the data with respect to the number of COVID-19 cases in the placebo compared to that in the active arm to see if the vaccine meets the efficacy standpoint. They will also be looking at the safety criteria. So they will be looking at the safety endpoints that are identified in the participants that got the vaccine versus the ones that got the placebo. And by having an independent committee reviewing this data, it takes away any possible bias in the conduct of this study. And that is really critical to the integrity of this clinical research and critical to public trust, because the public needs to understand that the clinical trials are independent and no one person or no one company will have a say into whether a product is deemed to be safe and effective. So this slide was taken from a paper that was published earlier this year in the New England Journal of Medicine, and it's broken down into two pieces. The top half of the slide refers to a traditional vaccine development. And in that, you have a traditional paradigm, which usually occurs over multiple years, or a regular infection. Initially, you have the target ID or the target identified, and then you look at preclinical information. Then you go into phase 1, which is typically the first in human studies, generally conducted in healthy volunteers. And you have a little bit of small-scale clinical material. Then you go into phase 2A that is looking at small efficacy data and trying to find the best dose before going into large safety and effectiveness trials, which are the phase 3 trials. And then you go into licensure. And companies typically don't get into large-scale manufacturing until they get into this last stage. And the big delay is going to be at the large-scale manufacturing. This, in the current pandemic, if we followed this analogy, it might take 10 to 15 years for us to have a safe and effective vaccine without changing the process. The key is to leverage advantages in the processes where we can without compromising safety. And I think that remains at the core of everything that we do. Now that we are in a pandemic, what would the vaccine development look like, or what does it look like currently? And let's look at the bottom part. So yes, you have the target ID that's identified. Instead of having the pharma company or an independent small biotech bear the full brunt and the fiscal responsibility or risk in case, because there is no proof that the vaccine will be effective, they have developed this partnership where the government has decided to invest in multiple different products to help speed up so that you can focus on the scientific process. You still have the first in human studies. So all the studies do have a phase 1 and an early efficacy trial. What it is you are really trying to expedite things by increasing the sample size so that events that might not normally take up to 5 years to occur can occur in a smaller timeline. At the same time, once a product is moving to the clinical phase, if it seems to be safe enough in humans, companies are already wrapped up with the manufacturing process so that when you have emergency authorization, I'm not saying if and when because we don't know when that will, it might be in a few weeks, it might be a few months. It will be when the science gives us the answers to those questions. But to then make sure that once you have approval, you actually have the vaccines to roll out in terms of capacity to help address the population. This is a scheme. On the figure on the right gives a brief description about the virus. So on the top, the semi-half circle that you see is the SARS-CoV-2 virus. And these spikes or crowns that are sticking out are called the S or the spike protein. The S spike protein has this S1, the bubble on the top, and the stem or the S2. And this whole thing is called the spike protein primer. The coronavirus uses this spike protein to then attach itself to the host receptor, which is the ACE2, angiotensin converting enzyme to receptor on the host cell. Once the virus attaches itself to the host cell, then it causes the receptor binding. It then fuses into the cell and then injects the RNA of the SARS-CoV-2 inside the host cell, thereby establishing the infection. This step becomes important because almost all the vaccine candidates that we are talking about are targeting this attachment and fusion and are in some way, shape, or form targeting this S protein spike of the SARS-CoV-2 virus. So what are the phase 3 clinical trial principles that are in place? They are all randomized placebo-controlled trials. So every single vaccine is placebo-controlled for the simple reason that currently there are no vaccines that are proven to be safe and effective against COVID-19. Therefore, at the current time, these are all placebo-controlled studies. Further, if you look at the sample size, they are all 30,000 to 60,000 participants. Never before have we enrolled participants in clinical trials that are this large because A, they are difficult to accrue, B, they are expensive to conduct, and it's beyond the reach of any one company to be able to do that. Almost all the phase 3 studies are currently over the age of 18, and they are including people most vulnerable to it. And what do I mean by that? Elderly people, are those who face the brunt of the pandemic, because when you look at the morbidity and mortality, they have been highest among the elderly. Those with underlying health conditions, heart disease, diabetes, COPD, etc., and minority populations, because these three have been the most vulnerable and therefore most impacted. So there is emphasis to ensure that these populations are included. What are the primary endpoints, or how do we say that a vaccine is effective? That's what we mean by endpoint. The primary endpoints for almost all the vaccines are virologically confirmed symptomatic COVID-19. And what do I mean by that? In order, when participants enroll in these phase 3 studies, they are followed over time for the development of symptoms. If they have symptoms, if participants in the study develop symptoms consistent with COVID-19, which could be fever, chills, cough, difficulty breathing, headache, etc., body aches, etc., they are asked to come into the site to get tested for the virus. And if they test positive, then they will be confirmed to have had a case. Okay. In addition, blood work is also done at various time points in these studies to look for the development of antibodies and to see, because we don't know how much antibodies you need to confer protection. At the same time, participants and study staff closely track any symptoms, because we want to make sure that these vaccines are safe. So all participants log on an e-diary or electronic on the phone app, their symptoms, whatever symptoms they have, especially the first week after the vaccine. And every week, study staff are in touch with the participants to ensure that we keep track of them so that we can make sure they have no side effects. And any symptoms that they complain to us, we report it and we assess that. Because we are blinded. We don't know. None of the study staff know who is getting the active vaccine versus the placebo. And it's our responsibility to report all the side effects without any prejudice to the database and ultimately let the data speak for itself. So we talked a little bit about the population, because you want to enroll people, you want to enroll, and we have enroll people who are at risk for not only getting complications from COVID, but also who might be at risk for getting COVID. That could be healthcare workers, first responders, EMS people, grocery store workers, transit workers, etc., because of the virtue of the work that they do, end up getting exposed and not necessarily being stuck at home doing only remote working. And in terms of the statistical considerations, most of the vaccines are counting cases 2 weeks after the last vaccine dose. The rationale for that is once you give a vaccine, it generally takes a few weeks' time for you to develop enough antibodies for it to be protective. So if you were exposed to COVID-19, let's say a day or two after you got the vaccine, it is unlikely that the vaccine will be able to protect you, because the vaccine hasn't had time to develop antibodies for it to protect you against the virus. Hence this timeline. And in general, almost all the vaccines require that they meet a minimum of 50%. That means they have to be at least 50%, if not more, effective to prove to the FDA that they are actually effective against COVID-19. So this is an overview, and this table may already get outdated because studies are really coming very fast. It's a snapshot of the various COVID-19 vaccine studies. And it's good to break them down by different platforms. So the first two studies, which are the furthest along, use the messenger RNA platform, and that's the delivery mode to deliver the antigen, the viral antigen to the host. The two companies that are furthest along are Moderna and Pfizer in combination with BioNTech. Both of these vaccines use mRNA, use 2 doses, and the Moderna has actually completed enrollment. They completed enrollment on October 23rd, and they've enrolled over 30,000 participants. And the Pfizer-BioNTech, Pfizer actually expanded it to 44,000. The second group is using an adenovirus vector, or using an adenovirus type that is not generally known to be pathogenic to humans. Both these studies, AstraZeneca and Janssen studies, were on brief hold because of side effects to 2 participants in AstraZeneca and 1 in the Janssen study. But subsequently, after review of the data, the pause has been lifted and they have been enrolling participants since then. The AstraZeneca is still at 30,000 and the Janssen, actually their sample size is 60,000. What's of note is Janssen is the only study of all of these that has only one dose. And that does have implications because when you talk about 300 million doses or 400 million doses, if a vaccine needs 2 doses, that means 400 million doses will only cover 200 million people. So that's why that's important. And the last 2 constructs are using a recombinant protein construct. And that's Novavax and the Sanofi in combination with GSK. Also both are looking at 2 doses. I anticipate that the Novavax will probably start by the end of this month and Sanofi in early December. So the messenger RNA, the first 2 vaccines that I mentioned on the table. The messenger RNA is a completely unique and different strategy because you're not working with the entire virus at all. You're really working with a very small piece of the messenger RNA that actually is responsible for coding the spike protein. What this helped is this has enabled early vaccine development, and which is very simple. And they took the messenger RNA that codes for the spike protein, used a nanoparticle delivery technique. And this, using this lipid nanoparticle, allowed targeted delivery of the messenger RNA to the antigen-presenting cells. There are 2 such products, the Pfizer-BioNTech 162b2, which is a 30 microgram dose, 2 doses, and the NIH Moderna mRNA 1273, which is 100 microgram, again, 2 doses given 28 days apart. I apologize for this slide if it's a little hard to read, but I can walk you through how quick some of the things have moved, in part because of the ease of the technology. January 11th, the very first time point on your left, is when the sequence was originally shared by the Chinese scientists. Within 2 days, the NIH and Moderna scientists had finalized the messenger RNA sequence for the spike protein, and by February 7th, in less than 30 days, they had prepared the mRNA 1273. That mRNA was then shipped to NIH, and in less than 2 months, they submitted that for an FDA. And by March 16th, which is just about over 60 days from when the sequence was first shared, the phase 1 study was started. And in the interim, the additional funding was given to the company to help speed up vaccine production. And within 2 months of start of the phase 1, the phase 1 data were made public. So by May 18th, it was published in the New England Journal of Medicine, and shortly thereafter, the phase 2 study was started. Again, phase 2 study was enrolled fairly quickly, and participants continued to be in follow-up. I will emphasize that participants in both the phase 1 and phase 2 study currently do remain in longer-term safety follow-up until data from the phase 3 study. So the phase 3 study was started July 27th and enrolled in just under 3 months. Enrollment was completed on October 22nd. So, and at this time, safety data are being collected, and it's the same thing as efficacy. So within a very short timeline, phase 1, phase 2, and phase 3 studies are ongoing and enrollment is completed. Even though the timeline appears to be fairly rapid, and it is. The DSMB has been looking at these data, and no serious safety signals have been identified. Therefore, participant follow-up continues to occur till this day. So this is a brief overview of the phase 1 study. In general, if you look at the color coding, the gray annotates mild, the blue is moderate, and the yellow is severe. And the bar graphs on the left are the participant side effects when they were observed after the first dose. And the bar graph on the right is the side effects after the second dose. In general, and the most common side effects here were arthralgia, fatigues, any local symptom and pain. That was the commonest. In general, participants who got the first dose had very mild symptoms, and those who got the vaccination, the second dose, it was generally, they had more side effects, but still it was mild to moderate, and very few people had severe side effects. So in general, and the severe side effects, if you can see, were seen in a much higher dose. So this dose, there's 3 grams at each thing. The topmost is a 25, the one in the middle is 100, and it's a 250 micrograms with the ones that got the severe. Based on these data, it was felt that the 250 microgram dose was probably the one that was causing the more severe side effects. So what are the immunology correlates? So the topmost graphs looks at the development of the receptor and the receptor binding domain for the antibodies. For the doses, I'm only going to talk about the 25 and 100 microgram because eventually with the 100 microgram that's been going into the phase 3 studies. So while you can see, even with the 25 microgram doses, there was some increases in the antibody titers. The titers were higher in those that got the 100 microgram dose, regardless of age. So you can see 100 microgram, 56 to 70 years of age, and compared to those greater than 71, it was still quite high. What's of note is on the bar, on this very first last bar or graph on the right, that's the convalescent serum. So they have compared the development of antibodies and the antibody titers in the vaccine recipients and compared that to the convalescent plasma because that is the closest correlate we have to what might be considered antibodies to natural immunity. Then they looked at various different platforms for neutralizing antibody titers. And so when you look at the graph on the right, as you can see, the neutralizing antibody titers were quite robust in the group that got the 100 microgram dose, especially after the second dose, because when you look at the x-axis on the bottom, you see essentially the titers are much higher after the second dose, after the 29-day. So it is clear based on this that these neutralizing antibody titers are quite robust. They occur and they are comparable, if not maybe slightly even better than the convalescent sera, which is on the extreme right here, as you can see. And most of the side effects were mild to moderate. Based on this, the phase 3 vaccine study was started, which is double-blind and placebo-controlled, including multiple different age groups, with the primary efficacy endpoint being symptomatic PCR-confirmed cases. Of note, the first interim analysis will be when 53 cases are confirmed in the group total. Because remember, the data are blinded, so we don't know in which group those cases are accruing. Within New Jersey, there are 2 sites, Rutgers, New Jersey Medical School, our site, and Hackensack was the other site globally. And just so you know, by October, and efficacy endpoints can only be accrued 2 weeks after the participant has received 2 vaccine doses. And if you can see, by October 23rd, over 25,000 participants had received 2 doses of the vaccine. So essentially, by now, a lot of participants have started coming into the period where if they develop COVID-19, they will be captured as a case or a primary endpoint for the study. This slide shows a brief overview of the diversity of the study population. The first graph depicts enrollment of minority population, 20% Latinx and 10% Black. It's not ideal, and I certainly hope we can do better with subsequent studies. When you look at the age group, there's a good split, as you can see, including about 25% over the age of 65 years. When you look at comorbidities, there's good representation with diabetes, severe obesity, cardiac disease, and lung disease, essentially the common conditions that people have. And these study analyses, really some of the interim analysis will be triggered when there is 53 cases. And that's only 35% of the cases. The second interim analysis will be 106 cases, or 70% of the projected cases. And the final analysis will be when 151 cases of COVID-19 have been diagnosed in this vaccine. So what does that actually translate? And I'm going to spend a little time reviewing this slide. So this slide looks at the likelihood of us finding the answer that we're looking at the various time points. The rightmost graph, or the graph on the with the dotted line, is the interim analysis. The dot and the lines in the middle is the second interim analysis, and the solid line is the final analysis. Okay, this vaccine was powered to detect at a minimum 60% vaccine efficacy, right? So if I follow this interim analysis curve, one, along to the point where it's 60% efficacy, and that's here, then the y-axis tells you what is the probability of me getting that answer if the vaccine is 60%. So this intersection here, where this primary analysis, the interim analysis one, intersects the y-axis, is about 10%. Therefore, what that means is, if the Moderna vaccine is 60% effective at preventing COVID-19 cases, then there is a 10% chance that at the first interim analysis, the vaccine will meet its efficacy. If it's less than 60%, for instance, what if it's 50%? If it's 50% efficacy, then there is, I can't even tell, it's close to zero. There's a slim chance, maybe it's a 2% chance that it will meet the endpoint. If, on the other hand, the vaccine is 80% effective, and I go all the way to the top, if the vaccine is 80% effective, then at the interim analysis, I have close to an 80% chance of proving that the vaccine is effective. So really, when people ask, well, oh, you have the interim analysis coming up, what are the chances that you'll find an answer? I don't know. It depends on how effective the vaccine is. The more effective the vaccine is, the more likely it is that you can prove that the vaccine is effective earlier. The second messenger RNA vaccine is the Pfizer in combination with BioNTech, or previously called the Oxford vaccine study, that also is looking at a similar messenger RNA for the receptor binding domain of the spike glycoprotein. This also was evaluated in phase 1 and phase 2 studies, and in general, the mean titers were quite robust. In fact, they were 1.9 to over 4.6 fold that of convalescent sera, so quite effective in reducing COVID-19 infections. When you look at the safety data for the phase 1 and phase 2 studies, the graph on the left looks at the local, and the graph on the right talks about systemic. You can easily see the bulk of the side effects were the yellow or the light green, so mild to moderate. There was very few in the local that had a severe, and none that had grade 4. Grade 4 is considered life-threatening. So there was no grade 4 adverse event identified in these studies. Systemic complaints also most commonly were fever, fatigue, headache, chills, and some muscle pain. But in general, across the dose, were found to be mild to moderate. So based on this study, the study population is enrolling 44,000 people. That's the target. And as of October, approximately 25,000 were enrolled. And the study was expanded to further, the age was included to include children 12 years of older, because as you know, that age group is a big driver of asymptomatic infection and therefore contributing to the ongoing pandemic. The planned point for the interim analysis is when 32 participants in this study will have symptomatic COVID-19. So that was the messenger RNA. The next two vaccines are the vector. So what do I mean by vector? It's like a Trojan horse. It's using a virus that's not pathogenic, a virus that does not cause infection to humans, using that virus to kind of carry a piece of the SARS-CoV-2 viral antigen and present that to the antigen-presenting cells, so that then it will teach the cells to produce the vaccine, to produce antibodies. There are currently two of those in phase 3 studies. One is the AstraZeneca, and the other is the Janssen. I apologize, I misspoke. The Pfizer-BioNTech was not the Oxford study. The AstraZeneca is the Oxford study. So I'm sorry for my previous error. There are other candidates, but I'm going to just talk about these two because those are the two studies being conducted in the U.S. The AstraZeneca uses a replication-deficient chimpanzee adenovirus. So what that means, it's an adenovirus that can cause infection in chimpanzee, but it's considered non-pathogenic, meaning it does not cause infections to humans. and therefore there is no pre-existing immunity, and therefore less likely to have side effects if given to humans. In addition, there is prior experience with using this similar vaccine delivery strategy for influenza, TB, malaria, and other viral infections, as you can see. Earlier this year, over 1,000 participants were enrolled and compared to Menactra, and 10 of those also received a booster to look at the role of the booster. When you look at the local and systemic adverse events, as you can see, in general, it was very well tolerated. Most of the participants reported mild symptoms, as you can see by this kind of light green yellow line. Very few of them had moderate symptoms. With respect to systemic side, and what was interesting is the top graph looks at the first dose, local symptoms, and the second line of graphs in the bottom looks at the second dose. It seemed to be much better tolerated with the second dose. And the same thing for the systemic complaints. Chills, fatigue, fever, feverishness, headache, etc. was much more seen in the first dose. And even those that had this moderate symptoms were much better tolerated with the second time around. When they looked at the immunologic responses to SARS-CoV-2, as expected, there was no immunogen, not significant anyway, shade in the comparator arm. And when they looked at the prime and the prime boost, the third graph, antibody titers in the prime boost were much more comparable to that in the convalescent plasma, showing that it was immunologically effective. Based on this, the AstraZeneca study is enrolling with the phase 3 study, using a 2-to-1 randomization, with, again, the primary endpoint similar to the other two studies, symptomatic COVID-19. I'm sure everybody has heard about the study pause that was implemented globally because of 2 participants in the UK who had neurologic side effects or transverse myelitis. Since then, to those who are skeptic because of the side effects, I would say the process worked the way it was supposed to work. When a serious side effect was observed, the study was paused. The independent data safety and monitoring board reviewed the data to see if it actually met, to see if participants, if there were other such side effects observed, and to make sure that the study was okay to proceed. Ultimately, it was felt that it was okay to proceed because the rate, it was not sure that it was necessarily because of the virus, and B, they could not prove that the rate was higher than would be expected in the population because transverse myelitis has been reported in the general population as well. The second vaccine is the Janssen vaccine, which I can tell you is, again, a replication-deficient adenovirus 26. And although it can be human pathogen, it's extremely rare and has been used for multiple other vaccines, Ebola, Zika, HSV, and in consideration for HIV also. It is using a similar pre-fusion stabilized spike protein for SARS-CoV-2, and it's looking at 2 cohorts of people, younger and older people. And they looked at single versus 2 doses. And when they looked at the immunogenicity of the vaccine between the 2 groups, cohort 1A was the younger group and cohort 3 was the older groups. As you can see, neutralizing antibodies, as shown by ELISA and in the other antibody testing, were found to be comparable to the convalescent sera. The dots on the green are the antibody titers that were seen in persons recovering from COVID-19 as a marker for what would be considered robust immune responses. They also looked at T-cell responses, and T-cell responses are helpful because even when antibody titers decline, T-cells have the ability to recognize and retain memory for long periods of time and therefore help the body. The phase 3 study did open on September 23rd, and Rutgers Johnson is a site for the study. There are multiple other vaccines, vector vaccines, and I just want to mention that there was one conducted from China looking at adenovirus 5, but that is a human pathogen, so it remains to be seen what the safety data on that are. And the one on the right, the Sputnik 5, where the famous Russian vaccine, as you will see, has already by Russian media data been approved. Although it's a small study with only 76 participants, and even though they had a good response, We have not seen any of the phase 3 data so without that, it's unclear to me how safe or effective the vaccine is. And the last section, which I won't spend too much time because these are just evolving, is looking at the role of protein and adjuvant subunit vaccines. So this is a detailed breakdown of the S1 and S2 pieces of the spike protein. And they include a full-length pre-fusion SARS-CoV-2 spike glycoproteins, and using these genetically identical protein and helping them bind with an adjuvant to help to teach the body. So they are essentially giving the protein of the spike protein, combining them with an adjuvant, and using the adjuvant to deliver this protein to the antigen-presenting cells that will then help the antigen-presenting cells develop antibodies in response to it. The advantages are that it has been, this technology has been studied. For instance, Sanofi has partnered with GSK to look at the role of this spike protein using one of their adjuvants that has been already approved for other vaccines, particularly the influenza vaccine. Phase 1 and 2 studies are ongoing, and if proven to be safe and effective, the phase 3 study will start in December. New Jersey Medical School will be a site for the study when the study launches. Novavax is the other study that is already will likely be starting their phase 3 study probably towards the end of this month. This uses a trimer using a matrix adjuvant that's present in the soap bark tree. And this pattern of combining this antigen with the nanoparticle is similar to what's been used in NanoFlu, which is a quadrivalent influenza nanoparticle vaccine, and the RSV vaccine for respiratory syncytial virus. This technology allows this combination of the antigen with the adjuvant to directly be introduced into the lymphoid tissue, where it's enriched with antigen-presenting cells, and also binds to the ACE3 receptors. And the adjuvant is necessary to develop robust clinical immunological response. And based on animal data, it appears to show fairly robust responses in animal models. With that, I want to conclude my talk with a shout out to my team and really the participants and our research camp and our various community partners that continue to help us do the work that we do. And this is our team motto. If you would like to reach out to us, that's research with the heart at ngms.rutgers.edu. With that, I will stop sharing to enable my colleague to present his slides.
Thank you so much, Dr. Swaminathan. That was very valuable, and we have a number of questions that we'll get to at the end of the program. And Dr. Gusmano, if you would like to go ahead and get your slides started, I'll just share that Dr. Gusmano is a professor here at Rutgers at the School of Public Health in the Department of Health Behavior, Society, and Policy, here again at School of Public Health. And he also serves as the Concentration Director for Health Systems and Policy. He's a research scholar at the Hastings Center and a visiting fellow at the Rockefeller Institute of Government. And his research examines the politics and ethics of health and social policy, with his research focusing largely on policy theory, and it explores two questions in particular. First, how should the public be involved in shaping public policy decisions? And second, what are the values embedded in traditional policy analytic techniques, such as cost benefit and cost effective analysis? He's the author of several books along with other authors. And from 1995 to 1997, Dr. Gusmano was a Robert Wood Johnson Scholar in Health Policy Research at Yale University. He received his doctorate in political science from the University of Maryland, College Park in 1995. Dr. Gusmano, I see your slides fine. And if you can unmute yourself, I'll turn it over to you. And thank you so much for joining us.
Thank you, Colleen. Well, thanks. It's very nice to be with you. I'm going to try to be relatively brief because I want to make sure that we have some time for Q&A and discussion at the end. But As you can tell from the really wonderful presentation from my colleague, Dr. Swaminathan, an enormous, an unprecedented amount of work has been going into the development and testing of a COVID-19 vaccine or multiple COVID-19 vaccines. It's really at a speed and a scope and with a level of global cooperation that we've really never seen before. Understandably, as she points out, this has raised a lot of concerns, but I do hope that the information she was able to provide you about the kinds of checks that are in place and have been in place are useful for giving the public more confidence in what's going on. I would say, and I had actually said this in another presentation, that I agree completely that the pauses that were put in two of the phase 3 trials were not bad news. Everyone was concerned when they read about this, but they were actually wonderful news, and from the sense of making sure that the processes and procedures are in fact in place and working to make sure that any vaccine that's developed and approved will be safe and effective. Now, having said all that, what I'm going to do is I'm going to talk a bit about COVID-19, but I'm also going to try to put this in a broader political and policy perspective and talk about how this fits in with the anti-vaccine movement and how some of the political concerns about what's going on, particularly in the US, although the Russian Sputnik 5 vaccine, which was issued approval before the development of phase 3 trial data were in, is an extreme version of this, but how those kinds of concerns also have the potential to undermine public confidence in a way that would really be unfortunate given the amount of important work that's going on to protect the public. So what I want to do is just sort of start out by acknowledging the John S. and James L. Knight Foundation and the National Science Foundation. I have two projects that are closely related in the sense that both of them are looking at issues around public trust in science and how we can improve public collaboration and public discussion of science, including vaccines. As you can see here, this is sort of well known to many of you. This is a slightly old poll, but we have had for a number of years now a large and growing anti-vaccine movement, which has resulted in a lot of public mistrust of safe and effective vaccines. So this is the MMR, measles, mumps, and rubella vaccine, which literally hundreds of peer-reviewed articles have provided evidence that these are safe and effective vaccines. They are extremely important public health tools that have helped reduce the rate of these diseases around the world. Yet you can see here from the Pew Research Center when they did a survey, parents of young children, a significant portion of whom were concerned about the risks of side effects from these diseases. This really started out in many ways around the world, or got kickstarted by an article that was published in The Lancet, which was later retracted, suggesting a correlation between these vaccines and disease, particularly autism, as a condition. And later they've been largely debunked, but various people, various elected officials, celebrities, have come out and tried to sort of raise serious questions about the safety and effectiveness of vaccines. This group, or these groups, have actually really been at the forefront of trying to raise concerns about the COVID-19 vaccines, suggesting that there's no way they could be safe and effective given the speed with which they're being developed. And this was, as Rand Paul put it, giving people a false sense of security. As you could tell from the previous presentation, this just doesn't fit very well with the facts, with the reality of how these vaccines are being developed, or some of the protections that are put in place. But nonetheless, this has sort of taken on a life of its own. Here is a global survey with responses from different countries around the world. And overall, right, the positive news is the majority of people who were surveyed when asked if a COVID-19 vaccine is proven to be safe and effective and is available, I will take it. The majority of people responding to the survey globally said that they would. But you see that the US is below the median. And so only 68% of adults in the US who were surveyed claimed that they would take the COVID-19 vaccine if it's proven to be safe and effective, which of course suggests that they're not really believing and trusting the government and the others who are involved in pursuing this research and creating the vaccines. And that is consistent with what we already know about the public response to vaccines. The good news is most people trust the safety of vaccines, 79% around the world. But then again, if you get down to North America and Western Europe, East Asia, Eastern Europe, you see far fewer people agree that vaccines can be trusted. So again, the concern about take-up is significant. If you combine this with the possibility, or really the probability, that any vaccine that is approved is not going to be 100% effective, that it may require more than one dose. As you saw, only one of the vaccine candidates requires one dose. The others are going to require two. So when you combine any concerns about the efficacy of the vaccine with the need for people to come out and get more than one dose and to follow a regimen, and then combine that with less than 100% efficacy, you have some real concerns about when we could reach herd immunity. And so taking on this issue of public attitudes, communicating effectively with people about why they can trust a vaccine is absolutely crucial. And this is undermining it. So forgive me, it says 2002. He hasn't been president quite that long. It's 2020. But President Trump, this is from CNBC, says no president has ever pushed the FDA like him. The vaccine is coming very shortly. I guess to his supporters, this is supposed to be good news that he's being aggressive and not letting bureaucratic red tape get in the way of vaccine approval. But to many people, this is a frightening prospect. He, on a number of occasions, suggested that a vaccine was going to be ready before a quote unquote very special date, implying that there would be a vaccine. Yes, I'm going to fix the date. That a vaccine will be ready before a very special date, implying that it would be ready before the 2020 presidential election tomorrow. And so this is clearly not going to happen, and leaders within the pharma community and the research community have shot him down. But it is suggested to many people that there could be a political timeline for the FDA approval of a vaccine. These kinds of statements actually prompted leaders of many pharmaceutical companies to come out and issue a statement suggested that they were going to follow safety and effectiveness protocols and would not pursue anything that did not meet them. And so you had an interesting issue of the private sector actually sort of countering a public employee. And this itself fits in with a much longer conversation, not just about vaccines and about treatments, particularly in the US, but frankly outside the US as well, where you have had a number of people who have tried to argue for lowering evidentiary standards used by the FDA and speeding up the timeline with which vaccines and treatments can be available. This would include, I would argue, sort of the calls for expanded use of emergency use authorizations for COVID-19 treatments like hydroxychloroquine, which later was sort of pulled, and the president's pressure on the FDA. So these are real concerns. And I would argue that furthermore, that they are also a reflection of a kind of larger war on science. One of the reasons why you see skepticism about vaccines and a relative lack of trust in the safety and effectiveness of vaccines by people in the United States is that there has been a significant decline in trust in all of our major social institutions, the presidency, Congress, but also science and technology. So people are skeptical about motivations, sometimes with good reason, but often in ways that interfere with public health goals. And indeed, we actually know that there is a recent history of rushing medical interventions in, and these are mostly sort of examples with regard to treatment. But for breast cancer patients, there was a rush to get in the use of bone marrow treatments, which later were proved to be harmful. In the book that I wrote with my colleague Karen Maschke from the Hastings Center in 2018, we talked about the cancer drug Avastin, which initially was used to treat advanced breast cancer and given approval on a fast track basis, only to have that approval revoked by the FDA later. Again, I would argue that this is good and bad news about the FDA. We heard about the protections that are going on at the clinical trial level, but at the FDA approval level, there are also important guardrails against making sure that we don't have public approval for vaccines or drugs that are not safe and effective. And indeed, the FDA throughout its history, despite the fact that it has often faced a lot of pressure, not just from elected officials, but various patient groups to approve things as quickly as possible and to not take drugs or vaccines off the market if there is subsequent evidence that they are causing harm that outweighs the benefits, the FDA typically has actually done so. And so we have within the FDA an important agency that protects the public's interest. And that's why it's been so frightening to many of us who observe the FDA and the governance of drugs and biologics to see the kind of unprecedented political pressure that's been placed on them. And when it comes to pandemics, this is not new. The sort of desire to get things done quickly, and one of the sort of leaders of an effort to develop a drug to combat Ebola early on in this pandemic raised some serious concerns about giving out untested drugs. So the protections that you heard about earlier for vaccine development are extraordinarily important, and it's important that we avoid the sort of over aggressive use of things like emergency use authorizations. Now, one question you may ask, why are people ignoring the facts when it comes to vaccines? Why are so many people skeptical of the MMR vaccine, which is clearly safe and effective? Why are they raising questions about COVID-19? One really comes out of cognitive psychology, right? So there is the phenomenon known as confirmation bias. When people have prior assumptions about things, they tend to place a great deal of weight on any new information that comes in that confirms what they believed before. And they tend to discount any information that they receive that is uncomfortable, that doesn't fit with their worldview. So if you're skeptical of government, if you're skeptical of industry coming in, you're going to look for any evidence. You might look for the pause in a phase 3 clinical trial and interpret this as evidence that this vaccine is going to be harmful. What you heard from both of us is that, in fact, it's quite the opposite, right? It is evidence that the process is working well. But it's important to understand why people are responding to evidence in very different ways. People also have different values. So there's been research looking at people who place great emphasis on cleanliness and purity and suggest that they dislike vaccines more strongly. And what's particularly concerning in light of our current political environment, particularly in the US, but in other countries as well, is a kind of tribal mentality and what happens when facts get caught up with one's self identity. I think if you look at the use of masks, for example, a pretty simple, straightforward public health intervention that, as far as I can tell, should have nothing to do with whether you are a Republican or a Democrat, a liberal or a conservative, and yet somehow has been politicized so that now people see themselves as mask wearers or mask rejectors, and this is very much tied up in their support for the current president. And so you have to make sure to better understand why people are responding the way they are responding to these kinds of facts as they are being presented. As my old colleague Daniel Callahan, the co-founder of the Hastings Center, where I'm a research scholar, suggested many years ago, is that facts are important, but they're not adequate, right? He wrote in the very first issue of the Hastings Center report, one of the leading journals in bioethics, that wise decision-making requires adequate and pertinent information. One needs facts, but what are the facts and what kinds of facts are needed? And this really gets to the heart of this concern about the role of values, right? Some of what we're debating about with regard to vaccines, generally a COVID vaccine in particular, has to do with the facts, right? What kind of immune response does the vaccine produce? What kinds of side effects and at what level of severity? But these debates often also get caught up with ideas about autonomy and rights, either the patient's autonomy. I should be able to try that under or untested drug if I want to, or I have a right to refuse treatment. The physician's autonomy and the right to make judgments about treatment based on clinical practice and public health arguments as well. Right. So when vaccine opponents talk about standards of evidence, they're not just being obstructionist, right? What counts as good enough evidence, right? This is what counts. What are they thinking when they're interpreting the evidence? One thing that is really important, and I'm going to sort of end on this point, and then we can go to discussion, is that presentations that give people information about the process for producing vaccines, evidence about their safety and their effectiveness are really helpful. But starting and just amplifying the presentation of facts is usually not going to be terribly helpful, particularly in the environment that we're currently in. So if you begin with myth busting, if you begin with science-based arguments, people are actually going to shut down. In fact, particularly the anti-vaccine crowd, but even those who maybe are not part of the hardcore anti-vaccine movement, but are on the fringes, are a little skeptical, would normally be open to vaccines, but are concerned, for example, about the speed with which you're doing it. If you just start calling them names and suggesting that they're ignorant or suggesting that they're anti-science, they're going to shut down immediately and stop listening to you, right? You need to pay attention and have a conversation about what they're worried about. In some cases, this may be about concerns about liberty or political power. My current doctoral student is writing a dissertation about the rejection of HPV vaccines among the mothers of young Black men. And one of the things they are concerned about is trust, right? You have even seen signs that have put up and calls to suggest that some African Americans are skeptical of participating in COVID-19 vaccine trials because they don't want to be guinea pigs for white people. This taps into a long history of discrimination against Black and brown people. The fundamental unfairness of the healthcare system reinforces those kinds of concerns. And those concerns are legitimate and they're real, even if the objection to a particular vaccine or a set of vaccines is misguided, right? You really need to engage with people and understand why they're afraid, why they're skeptical, why there is a lack of trust, right? So paying attention to the relationship individuals have with society, humans with nature, is simply crucial. And if we don't do that, it doesn't matter how much evidence we present. It doesn't how many facts we have on our side. It's not going to be particularly helpful. So I'm going to stop there because I know we only have about 20 minutes left and I want to make sure we have time for discussion. But thank you very much.
Thank you so much, Dr. Gusmano. That was great. And Dr. Swaminathan, I'll ask if you can perhaps allow yourself to come on camera as well. And thank you both. That was really well-rounded. Actually, that's giving us two important sides of this topic. There are a couple of questions I just will get started with. And I was thinking the first one was for you, Dr. Swaminathan, but really based on your last slide, Michael, I think I'll ask both of you to respond to this. And the question is about how do you communicate the science, right? A very technical vaccine, the different types that you shared, and how do you communicate that in a way to your communities for public health professionals in a way that, you can sort of help to make sense of what it is and make it understandable, and what message can you give? And I think in addition to that, Dr. Gusmano, if you can then add on, can you give some examples of language that you might use to try to address the things that you just mentioned? Maybe we can start with Dr. Swaminathan just on the vaccine types first.
So the simplest way to say is COVID-19 is a complex virus, and in our fight to try to find answers, there will be multiple different answers because no one answer will fit everyone. No one vaccine will probably be the right vaccine. There are multiple different studies, and we have to wait to see what these studies show and look at the evidence, because each vaccine may be slightly different, even though the studies are being done similarly. Some might, and I'm just hypothesizing, this is not proven, it's possible that some may be easier because let's say one requires only one shot. So if you live far off and need to come in, getting there twice, it's hard enough for you to have to take time off from work to come in for one shot. Getting two shots is just too much. So maybe that's that. Then it's also possible different vaccines may perform differently in different age groups or different underlying conditions. It's also possible that some vaccines may be more effective. I don't know, 80 or 90% effective, but maybe they'll have a little bit more side effects. Maybe you'll have a little more pain or soreness. And then, so you have to look at what the evidence at these trials show to then make an educated decision about what is right for you, because, our country is as diverse. I mean, the strength comes from its diversity, and there's no one thing that's going to be perfect for everyone. We have to all look at the evidence and then try to find the answer that best suits us. And, I would start from a place that, vaccines have been, have really helped change our lives. And I think we are the victim of our success because we have forgotten what it was like to have kids die or have serious complications from measles or mumps, rubella. These are serious conditions, kids paralyzed. I grew up in India. One of my classmates had polio. Till this day, she can't walk. She had to drag herself, with kids helping her to go to the bathroom. And she, it really, and to think a simple shot could have prevented that from happening is really a remarkable achievement. So, I think Dr. Gusmano put it perfectly. You can't really compact it. I think it comes from a place, anxiety about vaccines comes from a place of fear, belief, where your community, everybody in your family feels, and we are human beings. We are not robots. We can look at the evidence all we want, but we react from our heart and the way we feel, and change takes time. I'll let you add to this.
Yeah, no, I think those are all excellent points. What I would say is, I don't want anyone to take away the idea that I don't think facts matter or the science matters. So, for example, in response to one of the questions about the point of anti-vaxxers saying that there are other ingredients that have been used in vaccines as preservatives, as others that have caused potential side effects. That's right. I mean, that's really where Robert Kennedy, for example, I had his photo up, was coming from. But indeed, a lot of that has changed. And so the technology has evolved and there have been subsequent studies. And so I think sharing that is important. But in terms of how to approach people, I think as a general rule, active listening matters a lot. Some of you may have heard that phrase, but I think taking time to ask why people don't want to take the vaccine, what specifically they're worried about, right? It may be, for example, I've heard lots of people have talked about how long has this vaccine been in use? How long do the studies last? mean, one of the things that we're going to have to confront in an effective way is the fact that even with all of the advantages of the global cooperation in the development of a COVID-19 vaccine, if the phase 3 trial only lasts for 5 months or 6 months, you're going to know about the level of protection that it provides over 5 or 6 months. And we'll have to continue to track that going forward to figure out how, whether or not people need boosters or annual shots like the flu. And I think we can communicate that relatively effectively. We just have to anticipate those kinds of fears. With regard to one of the other questions about Black and Brown communities, I think it's a very complicated situation. I think first acknowledging the legitimacy of those concerns goes a long way. If you ask me in terms of a longer term strategy, no, I don't know. I think maybe taking on systematic racism would be a really good idea. And because it's not just Tuskegee and the aftermath of Tuskegee, even though that clearly continues to have an effect on people's attitudes, it's also about looking around about who benefits and who is harmed by the healthcare system as a whole, who is excluded from those kinds of benefits, and thinking, does this government really have my interest in mind? And I think unless you change the way we currently operate, it's really hard. It's much harder for public health professionals, for clinicians, for pharmaceutical companies trying to produce good products to convince the public that they should have their trust when it's happening within a larger context like that. So there's a short-term answer of active listening and being empathetic, and there's a longer-term issue of really taking on the fundamental problems we have that have undermined trust for decades. This problem didn't start with COVID-19.
Thank you both. The next question is there an issue if someone takes, let's say it's a 2-dose vaccine and they only take the first dose? Especially, and the other point is that if there are side effects that arise from that, it may be difficult to get them to come back for their second dose, which of course we see in routine vaccines anyway.
Right. So I think so when we look at the data, the reason most of the studies are recommending 2-dose because the first dose, for the most part, did not routinely give robust immunologic responses enough that we can measure to protect you from COVID-19. Ultimately, you're getting the vaccine so that you can be protected from COVID-19. In general, I would say most of the participants in the clinical trials had relatively mild to moderate. There were really none who had like severe side effects, right? And so I would use that to reassure them. But I would also, I think, preparing any person, like just any medication, clinical trial or not, before you give someone a medication, a pill, an injection, it's your responsibility as a healthcare provider to give them the full information that, you will expect a little soreness. You might have some fever. You will be a little headache. You might have some fatigue. If that happens, take some Tylenol. It generally will expect to last from, depending on the vaccine, maybe a day or two or longer. But preparing them upfront for that, so when that happens, they're not surprised and can take medications to prevent it so that they can come back for the second shot. Because at the end of the day, the reason to vaccinate or not, or to come back for the second shot, also depends on what you perceive as your risk benefit. If you believe deep down, facts aside, if you believe that your risk or the side effects that you're experiencing from the vaccine outweigh the risk of you getting COVID and having complications from getting COVID, you're not going to want the shot. Plain and simple. So.
Right, and as we point out in the article I shared, right, some of it's down to values and some of it's down to facts. And one of the things that you can do by having a conversation with people about what they expect is it can actually help to evoke the kinds of fears and concerns that they have and to address them head on. Early on in my career, we did a study looking at why older people agree to be vaccinated for the flu and pneumococcal disease. And it turns out there are many factors that contribute. The single biggest factor influencing whether someone goes for a flu shot has a medical professional ever told them it's a good idea. Dramatic differences simply by hearing from someone they trust that it's a good idea.
Excellent, thank you. I think we do underestimate the power of, especially when it comes from a healthcare provider, right? To get people to quit smoking, to change diets, and so on. One other question is regarding some information that I've seen that vaccines are required to be frozen rather than just refrigerated. And I wondered if one of you could speak to that. And in my eyes, that's a significant challenge for our local public health infrastructure that doesn't typically do that, doesn't have the resources to keep things like that frozen.
Right. It's obviously an even bigger challenge in lower income countries where this becomes a major problem in terms of having, the electric grid up to speed and in terms of, so yeah, there's a clear advantage if a vaccine can be kept refrigerated, but I believe that many, I'm going to turn to my colleague to figure out whether this is correct, but I believe many of those that are being developed will require freezing.
You're right. So it is challenging. So most of the vaccines do require freezing of some sort, most, but not all. That being said, once thawed, different vaccines can be stable at room temperature for 8 or up to 24 hours. So that would leverage a little bit. And which is why when I say there is going to be no one silver bullet, there's going to be multiple different vaccines that will need to be tailored to the community that you have based on the resources that impact your community. Because a community in Newark or New York City, the needs might be very different, but if you are in a rural kind of area with people have to travel far distances to come to you, or if you're going there to give immunizations, like in a van as an outreach. So there will be some that will be more stable and hopefully will not require that kind of refrigeration.
Well, and even some of the sizes that Dr. Swaminathan presented earlier about the sort of number of micrograms that are required and that are most effective are going to be really important because that could affect packaging, which is also going to affect manufacturing and distribution. So there are lots of small decisions that will have to be made that we'll only know sort of once we know which ones are moving forward. And that's why it's so extraordinarily important. And one of the things that's really different about this in Operation Warp Speed is the fact that they've put up money for manufacturing, as she explained earlier, at a time when there's no way most pharmaceutical companies would start manufacturing forget phase 3, before they had licenses or they were very far along in licensure, because it would be financial suicide to do that.
Thank you. One other question was specific to the mRNA format for the vaccine. And has that been used before, or is that something new? Because perhaps there's a perception that that's a very new approach to vaccines. So that leads to maybe some skepticism in the public.
Right. And sometimes only time will answer the skepticism. Sometimes you just need time and experience, because there are currently none that are actually FDA licensed for approval. Using that approach. That being said, it has, there is plenty of, clinical data based on this and other similar approaches in other disease entities, but we still don't have a licensed vaccine yet. And so that is something which is why these phase 3 trials are critical, because it would be important to follow them out safely as much as possible for the 2 years that the study is currently planned. Now, what will happen if the vaccine is effective, if there is an emergency use authorization because the vaccine is effective, it will start a whole set of new questions, because then you will have something that's somewhat effective. It also depends on what that means. An emergency use authorization doesn't mean it's a free for all, because a vaccine is going to be able to immunize the entire world. It's just not feasible. Mm-hmm. B, you just don't have enough vaccine doses of any one particular product. You're still going to need that. But I do think it's important that we be transparent about it, and that we be transparent about the process and how we come to those decisions, and to offer clinical trial participants certainly no less than what they would get as standard of care. Currently these are all placebo controlled, but that's because nothing is approved. the clinical trial people conducting the studies will need to reckon with what to do with participants who on these clinical trials have gotten placebo if vaccines are approved, and you're weighing that with the benefit of following safety, because you never want to be in a position where you ever withhold anything that's approved when you know that it's proven to be safe and effective. But I think it's important that we in the scientific community are transparent about it from the get-go to address any possible vaccine mistrust that may come just because we didn't do that.
And again, as I suggested earlier, and there are a number of examples of this actually with the FDA doing follow-up with the additional data being collected, even when things have been approved on a fast track, to come back and say that, well, subsequent evidence suggests that we need to revisit this. And it's going to be incredible. It's difficult to do that, right? Because once, if you read the hearing testimony that I read on Avastin, for example, you had lots of women, lots of families coming forward saying, this drug saved my life, please don't take it off the market, even though on a population level, right, there was evidence suggesting it did more harm than good. And so it took a lot of bravery actually on the part of the FDA to reverse its decision, but it's incredibly important, not just for these vaccines, but again, more broadly for the acceptance of vaccines. We're just incredibly important public health tools to make sure that whatever we do it safely and effectively and transparently.
And that is their job. And we, as a nation, certainly have come to expect that of them. And to see that potentially being undermined or undercut in any way is what throws everyone off, right? Is what throws and starts to lead to mistrust. Well, I believe I actually got to all of the questions. Is there anything else, Dr. Swaminathan or Dr. Gusmano, you'd like to add before we wrap up today's program? And thank you both so much. That was really valuable and informative. I just want to be sure we've covered everything that you would like to address.
I think, as I would just say, there are going to be multiple different vaccine candidates that will likely get approved over the next few months. Each vaccine will be different. There will be some nuances. I would just say that I don't think we should rush to make a decision about I don't think there is no rush to make a decision. Look at the science, make sure you get your facts from reputable sources so that whatever decision you make will be an educated one that's right for you and your community.
And I would just say, in your role as professionals, to make sure to take a moment to listen to the fears and not just immediately dismiss people who raise concerns about this, because it's reasonable for people to raise concerns. But if indeed things go well, there are, as you can tell, a lot of protections already in place. And so I think we can have confidence that the vaccines that are approved will be valuable tools for all of us.
Excellent. Thank you both so much for today. We very much appreciate your time, and thank all of you for joining us today. You should have seen that the evaluation link was placed in the comment box. I'll send that out again as well to everyone, but please do take the time to complete that. And stay well, stay healthy, and watch for information about some upcoming programs. Thank you both again for joining us. It's much appreciated. Have a great day, everyone. Bye-bye.
Thank you, Colleen. Thank you.